The second part of gene expression is translation, and this is when actual protein product is formed from the genetic code contained in messenger RNA. Recall that messenger RNA was created through the process of transcription in the nucleus. A portion of DNA called a gene was transcribed, creating a single-strand messenger RNA molecule that then leaves the nucleus and enters the cytoplasm to become associated with a ribosome. The ribosome is shown here in this gray circular form in the background. Now we also see a second kind of RNA called transfer RNA, or abbreviated tRNA here, that's important to the process of translation. Transfer RNA is the molecule that will actually bring each and every individual amino acid to the ribosomal complex for the construction of the protein. Now, transfer RNA is a ribonucleic acid, and therefore it is single-stranded. However, it folds back on itself in a number of places, actually hydrogen bonding with itself to form this very characteristic cloverleaf shape. One end of the transfer RNA molecule will hold the amino acid, a single amino acid, while the other end of the transfer RNA has what's called an anticodon. Now this is a 3-nucleotide sequence that will correspond with a codon on the mature messenger RNA. The ribosome that we've talked about is where protein synthesis actually happens. This is a large complex of ribosomal RNA, a third kind of RNA, and protein together. Now, two parts of the ribosome, a large and a small subunit, will clamp down on the messenger RNA as soon as it enters the cytoplasm. And there are two sites for tRNA to bind to on this ribosome, and I'm kind of showing them in a shadow form here at the moment. The first is the P site, and this site will hold the growing peptide. It'll hold the transfer RNA that's carrying the growing peptide as we go through this process. The second site is the A site, and that will hold each incoming transfer RNA that's carrying the next amino acid with it in sequence. Now remember, this happens outside of the nucleus. This is where we find the ribosomes. And of course, ribosomes, as we learned earlier, are associated with the rough endoplasmic reticulum. Remember, the presence of the ribosomes gives the rough appearance of the rough ER. Okay, so let's take a look at this process in a little more detail. In translation, what we see here is that the transfer RNA, after the process has started, the transfer RNA that has the growing polypeptide chain, remember this is what we're making through this process, is located in the P site. While the incoming transfer RNA that has the next amino acid in line is going to be arriving at the A site, this vacant site right here. And notice the anticodon on the transfer RNA corresponds, the CUA corresponds with GAU, the codon of messenger RNA. And that's how we get the specificity to make sure that each amino acid is placed in its correct place. Next, a peptide bond is formed between the current peptide chain and the incoming amino acid. So we have our two tRNAs bound to both of those sites at this point, and that peptide bond is formed through dehydration synthesis. And then the former tRNA that was in the P site now leaves. It is what we call empty. It has broken its bond with the peptide chain, and it now leaves, leaving the new tRNA that had just arrived to hold the growing polypeptide. And following this, the ribosome will advance down the messenger RNA, moving that transfer RNA into the P site, allowing another vacant A site for the next transfer RNA. Now, a functional protein is more than just a string of amino acids. It has to be folded correctly to be functional. And that's where the endoplasmic reticulum enters the story. So here in this diagram we see that the ribosome is this sort of pink blob here, and we see the messenger RNA is being held between the small and the large subunit like we've seen so far. And what we're seeing in this diagram is the ribosome being associated with the membrane of the endoplasmic reticulum. And you can see that the ribosome is releasing the protein that's growing here, because as the ribosome advances down the messenger RNA, we slowly add one amino acid at a time to grow this polypeptide, and you can see it enters the endoplasmic reticulum as it's being made. Now once it enters the endoplasmic reticulum, it immediately becomes manipulated by enzymes in the rough ER. And those enzymes will then fold that protein into its correct formation, making it a functional protein.